Good morning, everyone. Hi, everyone. Well, today I have the pleasure of introducing a scientist that I greatly admire. Um, Wendy Valencia-Montoya is a former student in the Department of Organismic and Evolutionary Biology here at Harvard, and is now a junior fellow in Harvard Society of Fellows. Throughout her career, Wendy has explored various questions and systems, combining fieldwork, phylogenetics, genomics, and physiology, probably among others, to understand what she considers the most beautiful process in the world: evolution. Her trajectory and insights have been recognized with numerous awards and distinctions and have also reached non scientific audiences through the pages of the New York Times. Her most recent work focuses on the interface between organisms and their environment, exploring the mechanisms organisms use to experience and navigate the world. The depth and richness of her work are such that it's impossible not to find yourself imagining how an octopus or a beetle perceive the world. It's a rare kind of science that sparks your imagination. But there's even more to imagine. Stemming from her commitment to understanding evolution, she takes the leap from studying phenomena at the scale of a neuron firing in a split second to showing us the millions of years of evolutionary history that have shaped the sensory systems. Today, we have the privilege of learning about an ecological relationship that has been in Wendy's mind since her first encounters with these organisms more than probably 10 years. So please join me in welcoming Wendy Valencia-Montoya.
Okay, thank you so much for that super generous introduction. It's not always that you get introduced by your close friends to a talk, so that's very sweet. And my project is about thermal radiation or heat as an ancient pollination signal. And it is about pollination. And as you probably know, plants have to mate and produce offspring while they are rooted in place. So the vast majority of plants, they need like some sort of agent to help carry their gametes around. And the way we think this pollination process started is by wind, like many ancient groups of plants still do today. For example, this pine tree is releasing clouds of pollen into the air. And this can be quite inefficient because you have to throw lots of pollen, hoping basically that it will land in the right place, in the right plant. But it usually, ends up in other places like your car, your clothes, your nose. So while wind pollination gets the job done, it can be quite a wasteful process. But then something extraordinary happened in the evolutionary history of plants, and it's that they started to interact with animals that could carry their pollen around. And a vision that will become very common soon is flowering plants with their stunning blossoms. And if you look at this, one of, I guess, most sublime views you get in nature, you may think that this has evolved to make us feel a little bit happy or less miserable in these like tumultuous times. But actually, they evolved to attract pollinators. And the evolution of animal pollination, that moment when animals and plants started to cooperate these ways, is one of the most transformative events in the history of life on Earth, I will say, because it made terrestrial ecosystems much more diverse and much more productive. And something that I was thinking about, because I know there's going to be pizza after, is just that imagining that you have a house and you are hoping for a pizza to get delivered to your house, and you call to the pizza place and ask for pizza being delivered, and then they just send randomly many pizzas in all directions, hoping that at least one pizza will land at your house. So that's quite inefficient. A better way is just to have someone that has directions to your house and that can carry a pizza, so you just send one pizza. And the way this happens in most ecosystems and in very complex ecosystems is by animals carrying pollen around. And one can study many aspects of pollination, and the way that I'm fascinated by is sensory systems, because it's trying to understand the signals and the sensory organs of the insects that help them get these directions to go efficiently to where they have to go. And perhaps one of the most striking features of thinking about sensory biology is the fact that every organism on Earth has a different sensory world. And we have radically different experiences of the same outside environment. So for instance, this is an image of what we humans see when watching this flower, while this is a reconstruction of what a honeybee sees. So a honeybee can see signals that are hidden to us, like these UV patches in the center of the flower that are guides for them to go to the center. I mean, this is an inflorescence, but like we can talk about these like flowers for this public. And the interesting thing is that this disparity in how we humans and bees perceive this outside world at the most basic level is due to the repertoire of sensory receptors that we have to make sense of the world. So the bee has an opsin, a UV opsin that we don't have, and this endows bees with UV vision. So for this example that I chose to introduce sensory systems, the honeybee can fly directly to the center of the flower, guided by this physical stimulus by light. And this light excites sensory receptors in specialized cells. And then these cells can convey this information through electric signals to the brain for the elaboration of behavior. And I was trying to study pollination not only from the interactions between the plants and the insects, but also very focused also on the pollinator, to try to understand these different levels of complexity, from the receptors that receive the signal all the way to behavior. So we know that the main pollinators are insects, and usually when we think about pollination, we think about bees, but there are also other groups of insects that we're going to talk more about today, and also others like birds and bats. And plants have evolved many signals to attract these pollinators. Some of these signals, such as shape, color, and volatiles, they have been extensively studied, while there are other signals hidden to human perception, such as infrared radiation, that they were not tested before, even though we know that there are some processes, such as thermogenesis, whereby plants can increase their temperature quite significantly above ambient. So for instance, this is a thermal picture of an aroid, and it can increase their temperature up to 46 Celsius above ambient. This is around 100 Fahrenheit, so they get very hot. And just by looking at how conspicuous are these cones and flowers under an infrared camera, it's easy to wonder that if pollinators had the ability to perceive infrared, this could be quite a reliable signal. And it is important to distinguish between only heat transfer by touching an object and infrared radiation, because if this water is hot, I cannot know, looking at it from a distance, only if I touch it. But having the ability to see infrared is basically perceiving these changes in temperature from the distance. And the conventional wisdom around thermogenesis is that it was useful to volatilize scents, but we challenged that conventional wisdom, and we thought that it could be used as a direct cue, because also we do know that there are some animals that can perceive infrared and use it for navigation. So for instance, snakes, they can form a thermal image of their prey, but also insects such as blood-sucking insects and mosquitoes, they use heat to try to locate victims like us in the forest. So then my question is if infrared has also evolved in mutualisms like pollination, and not only for in blood-sucking insects. So our question is if they are using this communication system, and as Danny mentioned, this project goes back many years when I went for the first time to this rainforest in my state in Colombia. And this rainforest is similar to the Amazon, like a tropical rainforest, but instead of being flat, the forest grows on this big limestone formation. And the cycad grows in these limestone formations on the top of these limestone rocks. And as you see, it's very complex, very crowded, and the beetles have to be able to find the cones, first the male cones, and then when they get to the male cone, they get all covered with pollen, and then they carry the pollen to the female plant. So in this complex forest, a cycad cone is sending signals that attract beetles, and then the beetles have very honed sensory systems to receive these signals and find their plants. And this interplay between the signals that the plants send and the sensory systems of pollinators is the base for communication. And as we all know very well, as anyone who has been in a relationship knows, communication is crucial if you want that relationship to last. And if this sometimes seems difficult to us in the scale of years, imagine now at the scale of hundreds of millions of years. And then think about the fact that this is one of the oldest pollination mutualisms that have evolved on Earth. So for more than 200 million years, descendants of these beetles and plants have been able to find each other. And this is quite important in this system because, as you probably know, cycads are dioecious plants. So they have male plants that produce male cones, and then they have female plants that produce female cones, or ovulate cones or pollen cones. And the beetles have to first find the male cones and then carry the pollen to the female plant when the plant opens. And then the plant has to be able to push away the insects and then close and continue the development of the seeds. And this can take up to 6 months because these plants are quite slow. So the beetles have to be able to pick the cones that are offering rewards. And they look quite similar to us, but not to beetles. For instance, we did this experiment when we have several different cones that they all look very similar. And we waited for beetles to arrive. And as you can see, a beetle arrived to the cone, to one of the cones in the middle here, and then is rapidly eaten by a lizard. I don't know if you see the lizard. And we didn't expect this, but something that got me thinking at this experiment is that it's not an isolated fact. When I look back to photos from the field, you can see that lizards are generally like close to the cones and are basically waiting for beetles to feed on them. As you can see here, all the lizards. So the beetles are under pressure to find their cones and to find them fast. And we were wondering if there were other signals, because we knew that scent and humidity are important, but we were interested in the role of heat, because we know that both male and female cones warm up when they are releasing pollen and when they are like receiving pollen. And the first question we asked is, how do cycads' cones heat up? So to answer this, we were interested in seeing the genes that were expressed in these different 2 stages, so the genes that were unique to when the hot is warming up. And this is how the graph looks like. So any genes that seem more abundant in this side, they are highly expressed in the thermogenic cones. And we observed that one of these genes, this mitochondrial enzyme, was one of the most highly and significantly upregulated genes. And this is an enzyme in the mitochondrial membrane that can deviate the process of producing energy to have a very inefficient respiration and just release the energy as heat. So usually what happens in the mitochondria is that we use sugar to produce energy, ATP, but what this enzyme does is that instead of using sugar to produce energy, it just releases this as heat. So we observed that plants, just by upregulating this enzyme, they are able to turn on thermogenesis. Something quite also beautiful that we found is that the tissues of the cones are full of these starch granules, like these amyloplasts, and that they get depleted after thermogenesis, suggesting that this is like the way they store the fuel for this very energetically expensive process, and also that these tissues are enriched in mitochondria, so you see a lot of mitochondria in the tissues that are producing energy. And apart from genes involved in mitochondrial function, we also found that there were genes that were upregulated that were involved with circadian rhythms, with like keeping time. And this was quite consistent with the data that we got from the field, where we were trying to collect temperature information from the cones. And when you record temperatures in the cones and also the ambient, you can see how they change. So you can track them over different days and also through different times. And what we observed is that thermogenesis is under a strong circadian control because you can see that they are always heating at the same time of the day. And then when we expanded this sampling across many other species, across the biggest radiation of cycads in the neotropics, which is the genus Zemia, we observe a very similar pattern, and is that they have this circadian control of thermogenesis where you see like one single period of heating. So we were interested in what the beetles were doing when the plants were getting hot, and we investigated pollinators of 2 different species of cycads. And we observed their behavior during thermogenesis, both by monitoring beetles getting in and out of the cones while we measured temperature, but we also had these experiments when we had like potted plants that we put in different groups, and we tried to observe how the beetles moved between these groups. And one way we went about doing this is that we have male plants that were separated from female plants, and we mark the pollen in the cones with UV fluorescent dyes, and then we could go and look with a UV lamp for marks of this pollen in the female cones. And then this will allow us to first see how much the beetles move, because you can measure these distances, and also at what time they are moving, and the parts of the cones that they are visiting. So this is an example of how cones marked with orange and green fluorescent dyes look like. This is one of the beetles carrying the UV fluorescent dyes. And this is like the marks that they leave when they enter to visit the female cone. And these are more photos from the field when you can see how the beetles are approaching the female cones. The female cones have these cracks around. And we observe that most of the beetles, around the cracks is when you see like more UV dyes accumulating, indicating that they mostly land there. And this was also interesting because we observed that usually around these cracks is where you see like a higher infrared radiation, so they are slightly hotter. And this variation in temperature in the cones was something that was not known before, and we observed it in both females and male plants, which is interesting because it's almost like a pollination guide. So this contrast between colors that point you to where to go when you land in the cone. And also with this experiment, we were able to reconstruct the timing of thermogenesis, but also when the beetles are in the cones. So we observed that most male cones heat up more or less with a peak around 6 in the afternoon, in the evening, and the female cones heats up much later, around 3 hours later, in around 9 p.m. And we observed that the occupancy of the beetles in the cones is highly correlated with these times when both the male and female cones are being visited by the beetles. And these experiments were very important for us to understand thermogenesis a little bit better, but because they were made with real cones, we cannot really disentangle all the signals that we know are important and are acting at the same time, such as humidity and scent. So we designed this other experiment where we scanned and printed 3D models of the cones, and this is how the cones look like in the wild. And then we yes, they look a little bit unfortunate, but this is what we got. And we artificially heated one of the cones, and we left the other cone at ambient temperature, and we did experiments in the wild. And as you can appreciate, the hot cone received many more visits by the beetles than the ambient temperature cone. And an important fact that is well reflected by this beetle flying here is that the beetles are making this choice prior to landing, because we covered the cones with glue. So once the beetles have made their minds and they have visited that cone, they were stuck. So they cannot just like touch and sense differences in temperature and choose between cones, but they only had one chance, indicating that indeed they were making this choice before landing in the cones. And we did experiments in the wild and also in cages where we control better for the transfer through convective heat, through touching, and through infrared radiation. And we observed that in both cases, the beetles were more attracted to the hot cones. Okay, so from this first part, we learned that through upregulating this mitochondrial enzyme, the cones heat up, and we learned that beetles care about this signal, that they are able to perceive. And then we wonder what are the organs that are involved with infrared sensing. And this was my favorite part of my PhD because it's the most aesthetically pleasing for me because beetles are incredibly beautiful. And their antennae is even more beautiful because they're covered with all these hair-like structures that they look like hairs, but they are the sensory organs of insects called sensilla. And underlying each of those hairs, there are neurons. And by seeing the shape of the sensilla, one can have an intuition about their function. So we observed that many sensilla in the last tip of the antennae, are consistent with a temperature sensation. And to check if indeed the antennae was the important part for this, we designed this simple experiment where we presented the beetle with a hot object, and we measured if they moved. So if they moved, we took this as a positive response. And what we did is that then we cut the last tip of the antennae, and we repeated this experiment. So with the beetles with their intact antennae, we observed that they responded to heat, and they also responded to plant scent that we use as a positive control. But if you cut the tip of the antennae, you see that you lose the response to heat, but they still respond to plant scent, and you can see that the beetle is missing the last tip, the last segment of the antennae. So now that we knew that, yes, this was probably where they have the organs to sense infrared, we directly measured the electrical activity of the neurons at the tip. And this experiment was quite difficult because these beetles are very small, only about 3 millimeters. But I have enlarged this beetle to make you feel that it was not so challenging. And the idea was to get electrodes to the tip of the antennae. And this is how the preparation looks like. So this is the beetle, the antennae, this is my hair, this is the hair of someone with a nicer, thicker hair. And you can see that it is very thin because it's just about the diameter of a hair. And we were trying to get to single sensilla. And we discovered this population of neurons that they were extremely thermosensitive. So this is a recording of a neuron. And all these lines is an action potential, so it's when a neuron is firing. And we found that when you increase the temperature, the spike rate, the activity of the neuron, mirrors the increase in the temperature. So this is like one of the most thermosensitive neurons that have been found, at least in beetles, and that could be consistent with a role in infrared perception. So now that we knew where the organs were, we wanted to understand the genes that underlie the perception of heat. And for this, we again tried to do transcriptomes. So I dissected like thousands of beetles because they're very small in different tissues to try to understand what were the genes that were highly upregulated in the antennae. So this is how it looks like. So these are all the different tissues, and these are different candidate receptors. And we observed that one of these genes, this one called TRPA1, the name is not very important, but the important thing to know about this is that it was highly expressed in the antennae. So the first step was to check if it was at the tip of the antennae, because we knew that this was the important part. And we checked for the expression of a positive control gene, that is this marker of olfactory sensory neurons, and we observed that indeed, there are many olfactory neurons in the antennae. And then we tested our gene of interest, and we observed that it was only expressed at the tip of the antennae, consistent with the function. And when we just mark all the cells that are in the antennae, we observed that indeed the expression of this gene is restricted only to the tip. So this was a good suspect for being the infrared receptor because of its localization. And the way we test the function of a gene outside of the beetle is that we try to put this gene in cell lines, and then we use a technique called patch clamp electrophysiology to record the activity of the cells. And I'm going to explain this because I didn't understand this before. So you have a cell, and the idea of this technique is that you are trying to gain access to it to record its electrical activity while you stimulate the cell. And if you don't have anything a protein that is activated by the stimulus, you shouldn't see anything, just like a flat line, not electrical activity. But then if you express your gene of interest, and you again trying to record what the cell is doing, and you stimulate, And if they are activated by these stimulus, these proteins should open and they should let ions inside the cell. And these currents will change the electrical charge of the cell. So it will create this, this inward curve. So we did this with our gene. We express it in cells. And then we increase the temperature. And we observe that they are very thermosensitive proteins because they also respond instantly to a change in temperature, very similar to what we saw with the neurons. And this was an important discovery because now we have a potential protein that is underlying the infrared perception. But usually, if you are working with model organisms, the next step will be knocking out this gene. But this is impossible because we don't have a genome. We barely assemble a transcriptome. So we were thinking about other ways of testing for this. And we thought about finding a blocker that is just a molecule that can literally sit in the center of the protein and stop its function. And after trying many, many different blockers, we finally found one that was very good for this protein. And we repeated the behavioral experiment. So remember that with a control solution, if we apply heat, the petals respond. And also if we apply the positive control, which is the plant scent. But if we have the TRPA1 blocker applied to the antennae, we observe that the beetles lost its ability to respond to heat, but they retain their ability to respond to plant scent, indicating that this seems to be quite specific for the function of this gene. And the last line of evidence that we found for the function of this gene is that, remember that we studied different beetles that pollinate different plants. So one of the plants heat up much more, up to 37 Celsius, while the other one heats up to 31 Celsius, more or less. And we also found that the activity of this gene was tuned to the temperature ranges that the plant, usually heats up, which is also another intuition about that this probably is the important receptor and that there is some extent of coevolution between the plants and the sensory systems of the beetles. Okay, I don't know how much time we have. We still have. Okay. So then, now that we have understood that this gene was important for infrared perception, we wanted to study how such a thermosensitive version of this gene could have evolved. And for this, we tried to compare our gene with one that was very similar. And they are indeed very similar because they only differ in the start of the protein. The variant that is sensitive to temperature is much shorter, so we just call this TRPA1 short and TRPA1 long. And this, if you map where is this in the protein, so this is the protein, this is like the pore where they carry like the electrical signals in, and this is just the small part that is different between the two variants. But that very small part plays a disproportionate role in the function because if you are missing that part, then this channel is very thermosensitive. But for the other version, then you increase the temperature and you don't observe a response. And this was also the same for the other beetle pollinator when you have the short version that is sensitive to temperature, whereas the long version is not sensitive to temperature. So to summarize this part of the talk, we found that this gene that is very thermosensitive is expressed at the tip of the antennae, and that splicing, like alternative splicing of this variant of the gene, can make a variant that is very thermosensitive and one that is not. Okay, so up to this point, we wanted to take a step back and think about how being able to sense heat and pollinate plants that heat up compared to other signals that we know are important for pollination. And we thought about color because it's one of the most important close-range signals for insects. And as you remember from the beginning of the talk, every organism has its own sensory world, and we knew more or less how will be the vision of a bee because we knew their repertoire of opsins, but we didn't know how this will look like for a beetle because we didn't have genomes for this beetle. But I went back and tried to look at where this was an important signal for them, thinking if they were using color, but as you can see, they all look very similar, so they all look like brown. But remember that this is how they look to us, so it may be that for beetles, they look differently. And so again, with the transcriptome that we produced, we saw that they have two opsins, a UV opsin and a green opsin. And this is interesting because they have lost one of the opsins, while the bees have three, they only have two. And then we mapped the colors of the cones. So we took the reflectance of the cones, the spectral colors, and mapped them in the space of vision of the beetles. I'm sorry, I think that. Oh no. Yes. So we mapped the colors of the cones, and you can see that they are all very redshifted, so they are around here. But the part of the spectrum where the beetles have the highest ability to discriminate between colors is here, where their two opsins overlap, suggesting that it doesn't seem that color plays much of a role for the beetles, and that they haven't differentiated much in the colors that they produced. And as you probably know, this is in stark contrast with flowering plants, because flowering plants have many different colors, and they have evolved to occupy all the possible range of the spectral colors. I'm sorry. And then what we did is basically to compare how these flower colors and cone colors look at two different visual systems of pollinators. And we compare with the bee because it's one of the most important pollinators. So while in the beetle you have two channels of information, two opsins to be able to see colors, in the bee you have three, and only having one more receptor gives you a bigger dimension of being able to discriminate, because here you can discriminate between millions of colors, whereas here you can only have like thousands color combinations, suggesting that indeed the vision of beetles is slightly more impoverished than the vision of a bee. But as I have discussed extensively in the talk, this may not even matter as much, because the beetles are probably not looking for color signals as much as they are looking for other signals like infrared. I'm sorry. And we know also that they have different repertoire of receptors, because while beetles have this very thermosensitive receptor, the bees have ancestrally lost this gene. So then this made us think like maybe there is a trade-off between these two different forms of signaling through heat or through color. And the way of doing this, of testing this in an evolutionary framework, is by gathering a lot of data for plants. And we had information about thermogenesis, but we didn't have as much information about color, because there are some parts of the plants that they have a lot of data, like Asteraceae or Fabaceae, but there are many other families that didn't have as much data. So to get around this problem, we developed an AI pipeline where we could gather color information from community platforms like iNaturalist, and then we could use GPT-4 vision to extract the colors from the flowers. And then using this information, we could then calculate some metrics of color diversity across basically many different families of plants. So this is a phylogeny of seed plants, and we first mapped the families that have thermogenesis. And as you can see, many of the plants that are thermogenetic, they are early divergent lineages of plants. And then we mapped color diversity, and we observed this pattern, and we tested and found that there was a negative correlation between thermogenesis and color diversity. So this basically means that plants that exceed thermogenesis generally show lower color diversity, which suggests that most plants are not investing heavily in both strategies. Instead, they seem to commit to signaling either through color or through heat, but usually not both. And this makes sense because producing heat is very expensive. We observed that they use a lot of sugar, but also producing pigment is also expensive because you have all the enzymes and machinery needed to produce these vibrant colors. Okay, and now if we step back again and look at the big picture, we tried to place all these findings on a timeline. So early divergent groups of plants that are pollinated by animals were mostly thermogenic. They produce heat. Whereas we see this deep time evolutionary shift toward plants that don't produce heat, but that are more colorful. And I was quite astonished when I mapped the diversity of different pollinator groups using fossil data, because what I found was, I don't know, I was just very struck by this, because there is extensive fossil evidence showing that the beetles were among the earliest pollinators to become diverse and abundant. You can see that the rise in beetle diversity precedes like the diversity of bees and butterflies, whose diversity really took off much later. And I feel that is not coincidentally around the same time. Around the same time, we start see the evolution of pollination systems based on color signals. So, and I think that one of the main contributions of this work is trying to help us rethink how the sensory biases, like the repertoire of receptors and their sensory systems that the insects have, how they have shaped the evolution of pollination signals, particularly this transition from metabolic cues like heat towards signaling more through visual colors. And it's basically like the community of pollinators that were around when plants were first starting to interact with insects and were starting to evolve these signals to attract them. Yeah. Okay. So in summary, I have uncovered the molecular basis of both thermogenesis in the cycad and heat perception in the beetles. And because my PhD was very long, like we do in OIB, there were similar findings that emerged in both systems while I was doing my work. For instance, we know that this enzyme also underlies thermogenesis in aroids and in water lilies. And also, we know that TRPA1, this channel, also underlies infrared perception in snakes and also in mosquitoes. And sometimes I wonder why to go through all the difficulty of finding the molecular basis of a gene. And to some extent, it's like going to one of the most basic levels of biological information. And then at this level, you can see these patterns of convergent evolution, how evolution has recruited, again, these same ancient genes to evolve these signaling systems, and how evolvable they are at the onset of just when a communication system is first evolving. And also how they sort of exploited this pre-existing molecular machinery that served as one of the earliest pollination signals for the attraction of insects. Okay, so this is the end of the talk. So if you're just going to remember one thing, let it be this. And is that even though today when we think about pollination, you picture bees and flowers and bright colors, pollination, like one of the most important interactions linking plants and animals on Earth, didn't start it that way, or at least that's why we observe, because among the earliest pollinators were beetles, and in the dark nights of the Permian and the Jurassic, they began searching for signals like heat or infrared, signals that we can't see or we cannot fully understand as extremely visual animals, but that are not less full of wonder. So they are probably were looking for signals like this, like hitting cones. And the irony is that I colored this red because we are very visual animals, so we couldn't understand that they were changing. Okay, so thank you so much. Thanks, Danny, for inviting me, and also to my labs at Harvard. And if you have questions, yeah, let me know. Yes. Sorry. Sorry, I think that's my phone. Sorry. Sorry. Yes.
That was really very cool to see. I had no idea that there was all of that heat treated pollination queue at all. I always thought about it in like the skunk cabin we have around here is just a way of beating the snow. I had a question. I was fascinated by the beetles just coming to the hot thing, like the experiment where you have the 3D printed cone. How, like, it might be that there's very few flowers throughout the forest that produce heat, but there's a lot of things in the forest that produce heat. How are the beetles not mistakenly going to anything that's vaguely
Yeah, that's a very interesting question and something that I'm working a little bit uh, on now. And is that we think about signals being useful at different scales. So for instance, scent is very efficient in the long range distance. And then once you get closer to be able to decide which cone to visit or where to land in a cone, then we think that heat becomes more important. And it's similar to the way mosquitoes do this. They get oriented by CO2 or scent, and once they come closer, they can use heat to see where to bite. So I think that one of the reasons this experiment works is because we have the cones close to plants where there is already like a sort of cloud of scent that can bring the beetles close to the cones. And also for the experiments with in the cages, we brought the beetles inside, so now they are using this as a short-range distance cue. So even though there are many other objects that are hot, something that looks like a cone and around the scent of a cone, then they are more likely to approach it. Yes.
So your pizza, of course, we're all thinking about pizza, and you sort of drove the talk in terms of insect pollination being a way of gaining efficiencies around male function. But cycads have some of the highest pollen-output ratios that you could.
One of the most what? One of the most what?
The cycads have very high pollen-output ratios.
Uh-huh.
So do you think that the innovation of insect pollination was really about male function and efficiency? Because, again, I mean, plenty of angiosperms have used very efficient pollination mechanisms, but cycads strike me as sort of pizza delivery in terms of pollen-output ratios, where they're throwing a lot of pizzas out there.
Yes, so that's a very interesting question because something that I didn't mention is that this is a case of brood-side pollination. So actually the beetles live on the cones and they feed on pollen. So rather than many other plants that they offer nectar as a reward and yeah, basically like sugar, and they carry the pollen but don't eat the pollen, these beetles feed on the pollen. And we also think that this is a very sort of early trait in pollination because many of the fossils that we found, they have the mandibles to feed on pollen. So they still produce a lot of pollen because they are feeding the insects with the pollen. But at the same time, they are making sure that they do their job. So it's like feeding your Uber driver, so they actually make sure to do their job well. And we also find this in other brood-side pollination systems.
So I guess what I'm really interested in your thoughts on is the origin of the pollination system. Again, there are two ways that you could look at it. Insects are feeding on pollen, and you're just losing pollen, and then plants essentially evolve away the cycads to put these insects to work. Or the first step is you attract insects to do pollination, and then you sort of let the system flow from there. So I'm really asking about the origin. Is it really about male function and efficiency, or is it something else? I'm just trying to think through the origin, not the sort of downstream effects, but the actual origin of insect pollination of cycads.
So I think that is mostly through male function because it is sort of retained that they produce a lot of pollen, but they don't have these sort of mechanisms to release the pollen. They actually have to open the microsporangia, like biting them, and they just access the pollen that way, and then they release it. So I think that the abundance of this first resource, like pollen, that probably they were just like coming to feed on something, and then this interaction got reinforced through, I guess, through evolution of signals like this. But yeah, I will think more about like male function.
A question about the synchronicity of this whole interaction, because you were showing this very like a stereotype way in which the plants produce heat, like it's a very specific time at dusk, right? How could the beetles come up with that, like synchronicity in their own circadian rhythm to get to those plants in the first place, right? Like are these beetles in clades of beetles that are already, they're active at night, or like how did in the first place they find the plants? Like how did that work?
Yes, so that's also actually something that I tested for this paper, and it's that most of the beetles that interact with thermogenic plants, at least in the tropics. In temperate zones, it's a little bit different, are nocturnal. And I think that this makes sense also because infrared radiation is a signal that is more effective at night when you don't have the sun and when the contrast is higher between a hot object. So I think that the fact that they were nocturnal, and also most of animals that are nocturnal, they don't have very elaborate visual systems, so this is also one of the reasons why they likely lost this opsin and they just get around with two opsins. So I think that there are sensory biases and behavioral biases that precede and make this interaction more likely to evolve.
How sophisticated do you think the beetle's heat sensing is? Because in sort of more visual systems like the UV bullseye, the actual shape is also very important. So like, is the actual shape of the channel in which the heat is strongest also important for them to be able to access it and detect it? Or is it just like for humans, like closing our eyes, just like, this is hot, this is not, or is it more sophisticated?
Yeah, that's a very beautiful question because it's like asking to step in the place of a beetle and how they look at this. So at least what we know is that this is important for navigations because they have these organs in the both antennae, and also they have at least two different like neurons that do this. And generally, when there are these two things in neuroscience, it's just to calculate it like distance. So at least we know that to finding distance and orient themselves like that. I don't know about the image forming part of it because at least we know from snakes that the organs that innervate the infrared sensor then meet with neurons from the visual system before going to the brain. So they are able to pull these two types of information before, so probably they are forming it's like a thermal image. But we really don't know how this works in the beetles, and I guess we will have to study a little bit more the circuits.
I have a question about the beetle diversity side of it. And just think about all of the beetles, like we've been, I think focusing on one species here. But what about the beetles that aren't feeding on pollen or feeding on carrion or something? Like, do you see the existence of this like thermoreceptivity confined to beetles that are seeking out plants that are producing heat, or do beetles that aren't interacting with plants in this way have the same receptors and are using them for other purposes?
Yeah, so we study at least two different species that they diverged more than 100 million years ago that were interacting with these plants, and they both have this very sensitive gene. But we don't know how this works in other species that are not attracted to thermogenic plants. But probably this receptor may not be as sensitive to temperature. But what people have studied is that there are many beetles that are pollinators of flowering plants and that are active during the day. And many of those groups have re-evolved opsin, the opsin that they lost, and have now gained, again, trichromatic systems, similar to how the bees see. So it really seems that the sensory receptors are changing very fast because of the pressure of pollinating in very different environments and according to the signals that the plants send. Yeah, so in summary, I know that this happens for the visual part, but we haven't investigated the thermal part in our species of beetles that don't pollinate thermogenic plants. Yeah.